So I think we'll begin. So again, good afternoon, everybody. Thank you for joining us. I'm Rosemary Collier, the person who's been emailing you all. So this afternoon, it's a great pleasure to welcome Professor Felix Wackers. And Felix is Director of R&D at the BioBest Group, who are global leaders in biological pollination and crop protection solutions. Felix's research focuses on the interactions between crops, beneficial arthropods, and the surrounding semi-natural landscape, with a particular emphasis on optimizing pollination and biological control. And today, Felix will consider challenges in conventional crop protection, and particularly, can biocontrol provide a viable alternative? So welcome, Felix. Thank you very much for joining us, and over to you.
Thank you, Rosemary, for the invitation and for the introduction. I will share my presentation, or will try to at least. Yeah, okay. As Rosemary was already alluding to, I'm going to, I think, based on my experience, and it's not just because I'm working in biocontrol, but I see that there is really a change happening at the moment in terms of open field crop protection. And that is driven on the one hand by concerns for the sustainability of farming, and on the other hand also due to the fact that there's a reduction in the options of conventional farming, and so conventional farmers are looking for alternatives. Let's see how. When we look at conventional farming, when I was at university, and that's already quite some years ago, we learned about integrated pest management. And that's a concept which is really has a lot of potential, and I really believe in it. And it's based on a combination of elements where you have first the agronomic practices. Then you have monitoring and the use of thresholds to determine if there's actually a problem that needs responses. And then if you do need to intervene, then you can do that in a number of ways. You can do that mechanically, biologically, or when everything else fails, chemically. That's the idea of IPM. If you look at the situation, and if you look at the situation that's often, not always, but often seen in the field, is that farmers only use part of these IPM tools, and often they actually prefer to intervene preventatively by using chemicals even before there is a real problem. And this is an issue because it means that unnecessary chemical interventions are taking place. And that in itself is also an issue because it not only kills the pests, but it also kills a lot of other organisms in the agroecosystem. And these organisms are important to actually stabilize the pest situation. And we'll see a few examples of that. If we look at the pesticide sales in the EU, even though there has been a lot of work and effort in developing IPM programs and developing alternatives, we see actually that the pesticide use over the last decade is more or less stable. There has been a trend in a slight reduction in pesticide use in the last couple of years, but it's really not what we would like to see. And that's the situation for the EU. If we look at other parts of the world, then we see that there's actually an increase, and sometimes a quite dramatic increase in pesticide use over the last decades. So we're not really going the direction that we would like to go. Having said so, there have been a number of trends in the last several years, which means that a number of pesticide options that growers have to control pests and diseases and weeds have been reduced, and due to a number of reasons. That's the reason that research has shown that some of these chemicals have side effects that were not initially identified. And there's also cases where the producers are retracting and taking chemicals off the market, not renewing the registration of these chemicals. And we see here a whole list of chemicals which have been retracted or not renewed. And below here, a number of chemicals which are in the process of being banned or not renewed. So there is really a number of important tools that conventional growers used to rely upon that are no longer available. So that's an issue for conventional crop protection. Then there's another group of chemicals which are being scrutinized at the moment, and it's chemicals which are based on PFAS, the forever chemicals. And it has been shown that these pesticides are, with PFAS in them, are a major source of PFAS contamination in the environment. And given the fact that, as the name forever chemical already says, these chemicals, once they are in the environment, it will be very difficult or impossible to get them out. So there's rightfully concern about these chemicals being used as well. So there's likely some more restrictions coming our way. And that's mainly a problem for open field agriculture, because open field agriculture, as I already mentioned, is still largely relying on chemical pesticides for crop protection. There's also agriculture of protected crops, so in greenhouses and tunnels. And here we already see for many decades that biocontrol has been successfully used in these crops. So they are not as much exposed to the bans of these chemical pesticides, but even there are some crops where there's going to be issues. Well, the argument from the chemical industry has always been that we do need these pesticides and in order to feed a growing population, and that is something which, to some extent, may be true, but I think it's also overstretched, this argument. Because if you look at those systems where we actually have by far the highest crop production, the yields, those are the greenhouse systems that I was talking about. In the greenhouse tomato production, for instance, you have 4 or 5 times more yield per hectare than you have in the open field, even though the use of chemical pesticides in many of these crops are minimal or they're not being used at all. Instead, we use biological crop protection, which works very well. So I think that by losing chemical pesticides that we would all starve, I think that's not the way that we should be looking at it. We should be looking at how can we develop solutions also for the open field systems that are alternatives to the use of chemical pesticides. And these alternatives, to some extent, are available because they have been developed over the last 100 years for these greenhouse crops. If you look at the sales of biologicals and natural substances, we've seen that the chemical pesticides are pretty much stable. We see that there is an increase in the sales and the use of these biological crop protection solutions. So that is good news. But as I mentioned before, this is largely being used in greenhouse crops. Examples of recently developed beneficials, that those are organisms that are present in nature and that are being produced and sold by companies like BioBest. And to address new issues, we also have to develop these new products. So examples here are hoverflies that are being used for aphid control, predatory strips on the right here that are being used for trips control, then some interesting mites here that are used for tomato russet mite control, but have been shown to be very effective also for the control of powdery mildew. Then there's Micromus angulatus, which is a ladybird species here, very effective to clean up aphids or as a curative solution. And then a brown lacewing, which is the Micromus angulatus, sorry, and the Propylea is the ladybird. The brown lacewing is related to the green lacewing, but differs in the fact that the adult eats the aphids as well as the larvae. So the question is, if biocontrol is so effective in the greenhouse systems, can we just copy paste this to an open field system? And I think that would be nice if that would be possible, but it's also too simplistic because there are key differences between the protected crops and the open systems. For one, as the name already says, the protected crops are confined and the open field is open. So if whatever you release in a greenhouse basically stays in the greenhouse, whereas in the open field, if you release your beneficials in there, and if those are beneficials that can fly or are very mobile, you have to make sure that they stay in the crop and that they're not moving on to the neighbor or somewhere else. Then in the open field, the biocontrol agents are exposed to the climate changes, climate conditions. Rain, precipitation is a big issue in open field, and we know that that can have a big mortality effect on pests, but also on biocontrol agents. Whereas in a protected crop, the organisms are shielded from these climate extremes. The temperature is standardized and so on. So it's easier to adjust your biocontrol system to a protected crop than it is to an open field crop. And very important as well is that when we're looking at these greenhouse crops, there's usually more budget to spend on biocontrol because these are high value crops. And in the open field, the value is often a bit lower or considerably lower. So that means that there are lower budgets to be spent for biocontrol in the open field crops. So rather than just copy pasting from the greenhouse to the open field, I think we have to look at new strategies to increase the efficacy of the existing biocontrol products. So that can be the existing products as they are being used in greenhouses, but it can also be biocontrol agents that are already present in the open field system naturally. And these new strategies can help to make biocontrol for open field systems more effective and cheaper. I always illustrate this with this example that we all know that Usain Bolt as a sprinter is extremely fast, but this man here on the right is actually faster. And the reason why he is faster, simply speaking in terms of muscle power, the reason why he's faster is that he has a tool to make his muscle power go further and more make it more efficient to move. And that's what we try to do with biocontrol as well. We try to develop these strategies so that we not just releasing biocontrol agents in a crop, but we make sure that we provide them with all they need to do their job. And if we look at a typical open field crop, and in greenhouses, that is also true to some extent, is that you have these large and increasingly large field situations where you have monocultures and there's a complete lack of floral resources. And these floral resources, we all know, are important for pollinators, for our bees. But what is maybe less known is that biocontrol agents are also entirely dependent on floral resources as a source of energy and protein, especially in those cases when there's no prey available. So what we can do is that we can actually reintroduce these floral resources into our agroecosystems by using them in field margins, and thereby provide these floral resources for the pollinators, but also for the biocontrol agents. And to show you that this is not just a trivial matter for biocontrol agents, here a study by Karen Winkler, who was a PhD student of mine in Wageningen. And she worked on the cabbage system where she had cages, large cages out in the field. In these cages, she introduced 20 cabbage plants. And in each of these cages, she also introduced the pest, which was the Tritella, the cabbage moth. In all the cages, she then introduced a single parasitic wasp that attacks this caterpillar. And in half of the cages, she added also a buckwheat flower for the parasitic wasp to feed, to get nectar from. And then she controlled the cages with the buckwheat flower, with those without the buckwheat flower. And you see the results down here. The two lines at the bottom are the cages, the two replicates of the cages without the buckwheat flower. And you see that in both replicates, the parasitic wasps lived only a maximum of 6 days. And in these 6 days, they managed to parasitize just a handful of caterpillars. Exactly the same situation, but now adding the buckwheat flower, and you see that the situation, the results are completely different. The parasitic wasps in the presence of the buckwheat flower survived more than 30 days each, and each individual wasp was able to parasitize more than 300 caterpillars during her lifespan. So you can see simply by adding these floral resources to the system, you have a complete different situation, and you really dramatically support biocontrol. Then you may say, okay, that's a parasitic wasp. Maybe you selected this because it's particularly dependent on these floral resources. Well, here's an overview of biocontrol agents that are feeding on nectar and or pollen, here in the last column. And you see that about everything that we currently depend on in terms of biocontrol agents are nectar or pollen feeders. So this is a very general phenomenon that we can actually enhance and support biocontrol agents by the provision of nectar and pollen. So if we go and introduce these floral resources, the question is always which flower mix, which flowers should we be using, how to optimize ecosystem services. And when I started with this work all the way back in the early 90s in the US, the idea was, as long as we increase biodiversity, then surely these biocontrol agents will benefit as well. I think we've seen over the last decades that that's not the optimal way of approaching this, because we're talking about completely different groups of organisms, and each of these organisms has different requirements, and especially pollinators and biocontrol agents are almost opposite in their floral resource requirements. So we're not talking about a one-size-fits-all approach, but instead we are actually looking at having your flower margins tailor-made to the ecosystem services that you want to support. And here to show you some of these detailed studies that would.
I was up at 6 because I had to check the sheeps, and then I've been doing assignment and like been on the farm all day.
Yes, here an overview of the type of studies that we've done to look at the question which flower sources are most suitable for biocontrol agents. And we've looked at attractiveness by doing olfactometer studies, and we've looked at nectar accessibility by having these studies where we look at different flowering plants. They are here arranged from open nectar to 4 being the nectar being hidden inside the flower. And you see that the flowering plants that are being utilized by all 3 groups here that we looked at, parasitic wasp, lacewings, and hoverflies, are the flowering plants where the nectar is very open and easily accessible. So they're really dependent on this for feeding. The reason being that these biocontrol agents don't have very specialized mouth parts, unlike honeybees or butterflies, for instance, that can actually have long tongues to reach deep nectar. Then, if we know that there's such differences in flower exploitation, then the question arises, can we also select flowering plants that are suitable for biocontrol agents, but not suitable for the pests that we like to control? And that's an important question to ask because some pest species are also nectar feeders or flower feeders. So we've done some studies here with the system, the cabbage system again, cabbage white as the pest and the Cotesia as the parasitoid of the cabbage white. And the blue bars here show you the responses of the cabbage white to a range of flowering plants. And you see that there's flowering plants here where the cabbage white is doing very well in terms of survival. And there's other plants where they are not doing very well, where they not seem to be benefiting. The control here is on the left. Interestingly enough, the parasitic wasp, the Cotesia, is not benefiting at all from those flowering plants where the cabbage white is benefiting. So if you were to select these flowering plants for your field margin, you're actually going to stimulate your pest problem, increase your pest problem, and you're not going to help biocontrol. And this is something which we were able to demonstrate in field studies as well. Luckily, on the other side of the graph, we also see that there's flowering plants which are very suitable for the parasitic wasp and not suitable or very marginally suitable for the pest. So we can indeed, in this particular case, select for flowering plants that support biocontrol and don't support the pest that you would like to eliminate or control. So we've done a lot of these lab studies or semi-field studies, but ultimately you want to make the step and go to the field and work with farmers and see if you can actually make a difference in a farming situation. And this is something which I have been doing over the last 20 years with field trials with farmers in the Netherlands, Belgium, Germany, UK, and Switzerland. So we have a lot of data, and a valid question to ask is, okay, you're putting out all these flower margins. What's the evidence that it's actually working? And I want to share with you some of the evidence that we have. So if we look at the flower margin here on the left, and you have a grass margin on the right, then you can compare the insects visiting these margins. And not surprisingly, in the margins where you have the flower margins, so the blue bars, you see that there's many more insects, including the insects that provide biocontrol, like hoverflies, parasitic wasp, and ladybirds. bumblebees are also clearly more prevalent in the flower margin than in comparison, the green showing the grass margin, the control, where there's very few of those insects. And we have done this over many years, and you typically see, if you're working with perennial field margins, that this difference becomes bigger over the years. So yeah, we see that these biocontrol agents are visiting those flower margins. That's not the same as having proof that they're actually feeding on those flowers. So can we also provide evidence of feeding? And to prove this, we have done work in the field where we collect parasitic wasps either from the flower margins or from fields next to flower margins. And then we analyze them by HPLC to see if they have raised gut sugar content. And this is work that I've done or that we've done in Europe, but also in the US, where we were asked to do work on bird conservation margins, which are highly diverse, naturally regenerative margins. And then we looked at a cotton field that was grown next to these bird margins. And as a control, we also looked at a cotton field which was bordered by Cahaba vetch, which is a single species, was planted as a single species, so low biodiversity. But we know that Cahaba vetch is very suitable as a sugar source for biocontrol agents because it has these so-called extrafloral nectaries, which are nectar sources outside of the flower on this case here, the bracts. And these extrafloral nectaries have evolved for the purpose of for plants to attract ants and other biocontrol agents, so they can actually, the ant can recruit bodyguards through the use of these extrafloral nectaries. So we collected parasitic wasp in the cotton field next to the bird margin, next to the Cahaba vetch margin. And this is the parasitic wasp that we concentrated on, Meteorus autographe. And what we see here is, first we looked at control individuals which had never fed on sugar, so they were actually close to starvation, and they had about 3 hours to live without the sugar feeding. The ones that we collected next to the bird conservation margin had sugar levels that were no higher than the control individuals that had never fed. So these parasitic wasps in the field were actually on the brink of starvation as well. And in sharp contrast, the ones that were collected next to the Cahaba vetch had more than a threefold in sugar reserves. So they had been feeding on the Cahaba vetches, and that Cahaba vetch feeding allowed them to become more effective, to live longer in these field conditions. Right, going on with the evidence, and now I'm glad to be able to present work from this project that I did while I was at Lancaster University. I worked together with David George, Pat Croft, and Phil Northing from Stockbridge and Farrer. And this project was called EcoStack and looked at the possibility of stacking ecosystem services in terms of agronomy and conservation. And you may be confused with another European project, EcoStack, that apparently got inspired by our acronym, and but it was later on, so that was in recent years. So in this EcoStack, we worked with a number of crops where we had the flower margin on the one side of the crop, a grass margin on the other side, and then we looked in the crop, collected insects, and did pest measurements 10 meters away from the flower margin, 10 meters away from the grass margin, and in the center of the field, which was about 50 meters away from both margins. And if you look at the results here, we've done that in a number of crops. Here, the situation with the aphids in peas. Let me briefly explain how you read this. So you have here the data collected next to the flower margin, here next to the grass margin, here in the center of the field. And each bar is a data point in time over the season. What you see that in the next to the flower margin, there are aphids on the peas, but they were not beyond the damage threshold, so there was nothing to worry about. And what is also interesting to see is that throughout the season, there's not an increase in aphid numbers. Actually, you see a decrease. Compare this with the situation next to the grass margin. There you see that you already start off higher with your aphid numbers, and you see a clear increase in aphid numbers as the season goes on. And how can we explain this? Well, for this, we look at the black line, and that's the amount of parasitism by a parasitic wasp of these aphids. And you see that there's clearly higher levels of parasitism next to the flower margin, and also earlier on in the season, it's already increasing. So this shows you that these parasitic wasps that are being stimulated by the flower margins, that are actually feeding on the sugars, as we have seen, move into the crop and do their work here in controlling the pests. And the further away you get from the flower margin, the less of this effect is visible. We've done this in other crops as well. This is here cabbage, where we looked at the cabbage aphid, which is a notoriously difficult one to control, but you see that the flower margins are doing the job. Again, much lower levels of cabbage aphid next to the flower margin as compared to the grass margin, and higher levels of parasitism. With the whitefly, cabbage whitefly, and caterpillars, we see a similar situation, even though with the whitefly, there's a short peak here in the season, but then it drops to very low levels, whereas it's overall higher next to the grass margin also for the caterpillars. And interestingly enough, in this crop, in this project, as there were a lot of industry partners involved, they also wanted to look at the yield. And I was afraid that we would not see any effects in terms of yield effects. But to my surprise, we saw actually in 2 out of the 3 years that we collected yield data, an increase in yield next to the flower margin in 3 out of the 4 crops that we looked at. So that was a pretty good result to have. And in the 3rd year, the difference were not significant. Peas here show a similar result in terms of the yield, more yield, 26% more next to the flower margin as compared to the grass margin. So that goes hand in hand with the elevated pest pressure next to the grass margin. Then there is another project which I started in 2004, and I think it's probably the longest running project in terms of this flower margin use, functional agrobiodiversity, and it's in the Hoekse Waard, south of Rotterdam. It's a fairly large island south of Rotterdam, and over the years we have seen that we started off with 3 farmers, and after 3 or 4 years, the farmers were so exciting that they themselves became ambassadors of the approach, and by now we have 97 farmers, which is a third of all farmers utilizing this approach. There's a waiting list actually of farmers who want to participate. And we have 800 kilometers of flowering field margins in this area. Most interestingly, though, is that we've seen a 90% reduction in insecticide use in cereals and potatoes as a result of this approach. And I think that's a result which is not only attributable to the flower margin, but it's also attributable to the fact that growers that participate in this project do actual proper monitoring. And I think the monitoring is one important element to reduce the unnecessary pesticide use. So in summary, we see that these large-scale projects increase biodiversity, they help reduce pesticide use, and in certain cases can increase yields as well. So this is one approach that you can take. It's not always possible, or not all farmers want to sacrifice some part of their land, even though it's really not much in terms of percentage, to flower margins. And so in those cases where you don't have these flower margins, there's also another possibility, another way of providing these food sources, the sugars or the pollen. And that's by actually applying the food supplements on the crop itself. For this, we developed a pollen product, Nutrimite, which is actually based on Tifa angustifolia pollen. And that can be dusted on the crop, and we will see an example of that later on. Sugars can also be sprayed on the crop, even though there you run into potential issues with sooty molds. So you have to do that in a particular way to avoid that. And there's also other food supplements that you can use, like fictitious spray, and artificial diets could be used as well. So let's look at the pollen. Pollen is an important food source also for a number of biocontrol agents, especially predatory mites are notorious pollen feeders. Hoverflies need pollen to mature eggs. Without pollen, hoverflies will not lay a single egg. And predatory thrips, which we're working on at the moment, are also well-known pollen feeders. So I'll show you some results here of a trial that I've been doing for the last 4 years in onion. This is south of Antwerp. You can see the harbor here in the back. I live myself in Antwerp, so that's convenient that it's next door. And this is a conventional onion grower. And in onion, there's always a big issue with onion thrips. And these onion thrips can do severe damage. So what growers do is they spray against these thrips 2, 3, 4 times a season using various insecticides. I walked up to this grower and asked him, you can please do whatever you are doing normally, but in the corner of your field, can I just apply pollen and see if that is a way of actually supporting the natural enemies in your system? And he was very open to this and allowed me to do so. So that's what I did. Then I monitored for the onion thrips and monitored for the natural enemies as well. And natural enemies predominantly were these predatory thrips that we've seen before, but also spiders and lacewings were often seen. So if you look at the number of biocontrol agents, so natural enemies in the field with the pollen only, that's the orange bars, and compare that with the conventional treated crop, then you see that there's many more biocontrol agents in the pollen treated part of the crop, and only very few biocontrol agents are found in the insecticide treated crop. Well, that's understandable because these insecticides do also affect the natural enemies. And on top of that, we feed the natural enemies in the part where no pesticides are being used. If we then look at the onion thrips, and that's what the farmer is mainly interested in, then we see here 4 plots where the pollen was applied and compare this to the situation in the rest of the field where pesticides were being applied. So we see for each of these locations 4 monitoring in time. We see with the conventional crop that the pesticides are doing their job. They are reducing the onion thrips significantly compared to the pollen treated part where no pesticides were being used. but you never kill all the pests, so there's always some pests surviving. And the ones that survive in an environment where all the natural enemies are gone, so they can then quickly rebound and develop large populations. And here at the end of the season, the trips damage really got out of hand. We had more than 60 onion trips per onion plant. Compare this now with the situation where the pollen was applied. You see that we have initially higher levels of onion trips, but still below the damage threshold. And what is interesting to see is that even though there's no pesticides being used, and there was also no drift, that the onion trips actually doesn't increase. It stays stable between the first two, or slightly reduces between the first two monitoring time slots. And then we see that it completely collapses the population of onion trips. So even in this last day when we saw these huge numbers of onion trips in the chemically treated part of the field, there's hardly any onion trips left in the pollen treated part. And this is how it looked like at the end of just before harvest. You see that the conventionally treated crop is completely damaged, burnt by the onion trips, whereas the part where the pollen was applied is green and thriving. And the farmer was saying, if my whole crop had looked like the green part here in the corner, I would have waited another 2 weeks to harvest, but there's no photosynthesis going on here anymore, so I'll have to harvest. Then we also looked at the yield. So I measured the diameter of the onions as well as the weight, and we saw that the diameter was 8% larger in the pollen-treated one part of the field, and the weight was 15% higher. So this shows you, I think that the two things combined, getting back to this question, do we have to accept lower yields when we stop using pesticides? That is definitely not always the case. You can have very high yielding situations where pesticides are reduced, have been reduced, or even completely omitted. A third topic here, but I see that we're running out of time, so I'm going to skip this one. It's ways of dealing with ants, which can be a problem in biocontrol, and you can do this by providing sugar sources that distract the ants. And that works very well. So you can see here that in grapes or in apples or in citrus, that this works perfectly. Here, work that we've done in apples in the Netherlands, where you see that just by using the sugar feeders, the ants are distracted from tending the aphids, and the aphid populations collapse completely compared to a situation where you don't do this. The last one is another resource. We've been talking a lot about sugars and pollen, but there's also other resources that you can use to support biocontrol. And one thing that if you look in nature, you see that a lot of plants and trees have these kind of hairy tufts under their leaves. And you may ask, what are they for? Well, if you look a little closer, then often you see that there's predatory mites sitting in these hairy tufts, which are therefore called acarodomatia, so little houses for mites. And often you see also that these mites use these hairs to lay their eggs. So a number of mites, predatory mites, are completely dependent on these hairy structures to survive. And if a plant doesn't have these hairy structures, they don't establish or they don't survive in these crops. So if you look at the crops where this is an issue, then for instance in pears, there's no hairs on the leaves. Some grape varieties don't have hairs, citrus, roses. So these are all plants where we would have problems with establishing biocontrol agents. So we did do some work with citrus to see can we actually introduce these hairs artificially, and what effect would that have on the biocontrol. So first we selected a fiber, a natural fiber that was particularly suited for this purpose. And then we did a trial where we either provided no resources, we provided one resource, so the fibers, pollen, or sugars, a combination of two of them, for instance, fibers and sugars, and a combination of all three. Then we treated citrus plants like this, introduced predatory mite Azaeus stipulatus, and looked at the development of this mite on the citrus plants. Control up here, you see no mites established in the absence of these resources. If we have one resource that we provide, we see they are at least establishing, but the populations are not very impressive. Combine two, then you see that especially when there's fiber and pollen combined, the mites are building up very high populations. Combine all three, and you get by far the highest population buildup. So this is a clear example of a synergy where you, by providing multiple resources, can actually get a situation, not 1 + 1 is 3, but as we had 3 resources, we saw a 30 times higher establishment of these mite populations as compared to a single resource. And I think, yeah, these are a number of examples of what is called conservation biocontrol. So we actually alter the crop or alter the landscape in a way that actually supports the biocontrol agents. And in my opinion, there's still a lot that can be done in this field because we have been always looking just at this nectar and pollen, but there's so much more that you could do in terms of conservation as well. Think of mulches and so on. So this is definitely a worthwhile field to be working in for you, those of you who are starting off in research. That might be an interesting topic to look into. And I thank you for your attention, and I've left 10 minutes for discussions. I'm happy to hear from you.
Excellent, Felix. Thank you very much. And a lot of food for thought, literally. So has anybody got any questions? You can ask them in person, or you can obviously type them in the chat as well. Right. Okay. Marta, would you like to ask in person?
Yes, thank you very much, Felix, for this interesting presentation. I was wondering about the flower strips, which is super interesting. But in terms of the glass houses, you mentioned that usually, you don't experience maybe many problems with pollinators or when you apply biocontrol in a glass house. And I know one of them was environmental conditions, but do you also in the glass houses need to have some flowers or something to kind of maintain those biocontrol agents, or does it apply only to the field? Is the flowering of the cultivated plants in the greenhouse sufficient?
A very good question, actually. And I think in the greenhouse you have to distinguish between types of crops. So you have crops like the greenhouse vegetables that typically flower. And if you look at a crop like tomato or bell pepper, they flower throughout the cropping season. And in those cases, especially with bell pepper, they provide nectar and pollen directly. So in that case, there is not necessarily as much need to apply these resources. In the case of tomato, for instance, it only produces pollen and doesn't produce nectar. So there is a possibility to enhance biocontrol by the use of nectar plants. In the past, we've had what is called banker plants being used already. So that's an old idea. And in these banker plants, there were aphids they were inoculated with aphids that were not able to transfer onto the crop. For instance, wheat aphid doesn't go on peppers. And then these banker plants were used to produce populations of biocontrol agents. Nowadays, we see that there's also flowering banker plants being used. So not necessarily with aphids, but with floral resources. And lobellaria is an example of this, and that's being more and more being used in greenhouse crops as well. What is also interesting to see is that this idea of flower margins is now being tested around on the outside of greenhouses to see if we can stimulate biocontrol agents there, and hoping that they would move into the crop, and as well as inside the greenhouses. And this is a good example where actually the open field systems have been ahead of the greenhouse systems. So the greenhouse systems are now copying what we have been doing in open field systems for many years. And it's good. Then we can learn from both sides. We can learn a lot about the use, augmentative releases of biocontrol agents in greenhouses for open field systems, but also the other way around. What we're doing in the open field is often also relevant to greenhouse systems.
Thank you.
Thank you. Right, there are quite a few questions in the chat now, Felix.
I see.
Yeah.
What I already was mentioning, should we not be able to go through all the questions right now? I will try to answer them by email. So let's see, where do I start?
So Harriet, I think, was first. So Harriet asked, how far do the biocontrol products travel, please?
Yes, that's a good question, important question as well. It depends on the biocontrol agent, on the organism. So the hoverflies, for instance, they are very mobile. They can fly over hundreds of meters. They actually migrate from Spain all the way to the UK every year. So that's an extreme example. And then there's other flying organisms that can be moved by the wind into the crop, also over big distances. The non-flying biocontrol agents, like the predatory, the ground beetles, they are typically not quite as mobile, even though there's examples as well of ground beetles that can move over hundreds of meters. But typically, I recommend to have these flower margins for a width of a field of 75 to 100 meters.
Okay, thank you. Next one's Hannah. Could the application of additional resources, e.g. pollen spraying, be used to increase the efficacy of flower margins further into the field?
Yes. That's a very good question. And actually, with this example of the onion field, you couldn't see it, but there was a flower margin where I was taking the picture. So the flower margin was going along the pollen treated part of the field as well as the conventional part of the field. And the idea indeed is that these biocontrol agents, like the predatory trips, you see them in the flower margin first, and then you use the pollen to actually move them from the flower margin into the onion field, where they can do their work in controlling the onion trips.
Okay. What's your biological recommendation for field potato beetles?
Yes, that's that's a difficult one, given the fact that Colorado potato beetle is not native to Europe. It's originally from South and Central America, and when the potato was brought over, at some point the Colorado potato beetle was introduced into Europe as well. But typically when you have invasive pest species, it's the pest coming in and the natural enemies staying behind in the country of origin. And that's also what happened with the Colorado potato beetle. So they are here in a what's called an enemy-free space. so they can cause serious problem. My biological recommendation for Colorado potato beetle is to make sure that you don't plant near a field where potatoes had been grown the year before, because they tend to overwinter and come out of the soil in spring and then start walking to find new fields. So if you plant your potatoes next to a field where there were potatoes with Colorado potato beetle, then you will have problems. Field margins will have a positive effect also on the Colorado potato beetle, because it stimulates some generalist predators as well, like predatory bugs and spiders that can have an impact on the larvae of the Colorado potato beetle.
Great. Next one is, what's the relative cost of these beneficial food sources if applying to open field crops, and how much needs to be applied to be beneficial in maintaining the populations?
Yes. The amount that you need on a hectare is not really very high. We apply, and we have studied this, that 500 grams per hectare is enough to actually do the job. And in the open field, you also have the advantage that you can use the wind when applying this pollen. So you walk upwind from the field, the pollen drifts over the crop and settles on the crop over a longer distance. So it's easy to apply. The cost of this pollen product is about 90 to 100 pounds in your case, or euros per bottle. So that means if you apply it twice, what we recommend, the cost will be about 200 euros or 200 pounds per hectare, which is really competitive when you compare it with the cost of pesticide applications.
Great. So there's somebody, next person, thank you for your presentation, and asking whether spraying with fiber would reduce photosynthesis.
No, because the fibers that we used are in the spray application. It's just individual fibers, so it's enough if one or two fibers hit a leaf, and it doesn't interfere with the photosynthesis at all. There's another way that you can use this as well. That's by putting a kind of a cloth or a netting over the crop, and that can have an impact on photosynthesis.
Great. Then the next question was about, is pollen sold? But I think you've already said that it is sold commercially for growers to use.
Pollen, yes.
Yes, yeah. Then one about, again, thank you from Michelle. Tea Piri is good on apple, presumably because of hairy leaves, but andesonite is useful on cherry with glossy leaves. Presumably different predatory mites require different amounts of hairiness.
That's absolutely correct. So there are predatory mites that will never establish on smooth leaves, and there's ones that don't seem to be dependent on these hairy structures, but that's just one or two. So from the ones that are commercially available. So you really restrict the number of predatory mites when you don't have these structures available. And given the fact that each of these predatory mites has their advantages in terms of climate adaptations and so on, by restricting it to one or two species, you really run into problems. And we see that in a lot of ornamental plant species as well in greenhouses, that biocontrol doesn't work very well due to the fact that these plants have these hard and glossy leaves.
Great. Next one is about, you might have to read this one to understand it. Pollen application, has this been used to boost rubus and other T. fusipenis controlled by allothrips? If so, how and when applied for best effect?
We've so far only done work in in onion and in strawberry. In strawberry, because in those two, we know that this aiolotryps is naturally occurring. So they seem to have an affinity with those crop plants. Doesn't mean that they couldn't be important in other crops as well. In rubus, is mentioned here. I know that in rubus they're also present. So it's something definitely to look into. It's a new product for us. We're in the research stage. So looking at additional crops is one of the things that we would like to do.
Excellent. Next one is any planned trials for Almeria and the aphid increases that have been seen there again this season, peppers and tomatoes. Where could they get the beneficial insects from in Spain?
Well, we have a distributor in Almeria as well in Spain. And I know that in Almeria, actually, I'll be heading off there in 2 weeks to discuss the trials for this season. In Almeria, there is an issue with the aphids, but an even bigger issue in Almeria peppers are the Thrips parvispinus, which is an exotic thrip species, which really causes havoc in pepper cultivation as well as in berries. So we're looking to test a few new biocontrol agents to see if they can help in terms of controlling these pests. And the food supplements are going to be part of those trials.
Excellent. I think we'll just go with one more question because we've got to 5 past. So I'll go to this one here. Silicon applications have been shown to enhance trichome production in some crops. Have you considered trials combining silicon and biocontrol applications?
No, we haven't looked into that. I'm indeed familiar with that, with those papers. So it's something which could be interesting to look into. And the question is, if it enhances trichome production, does it mean that the plant needs to have already trichomes and then it produces more, or would it? I think it's unlikely that a plant that doesn't have any trichomes would all of a sudden start sprouting trichomes by applying silicone to it. But it's something, definitely a good recommendation, something to be looking into.
Great, thank you. So there are a few more questions and comments and thank yous, which I'll send to you because I don't think you'll probably be able to access the chat. And I mean, you've put your email address on there, Felix. So are you happy for people to email you with questions?
Yes, that's fine. If you have particular questions, you can always reach me there as well.
Yeah. Okay. Right. So I think we'll stop there. So thank you so much, Felix, for a really, interesting and informative presentation, and say lots to think about. You can see from all the questions as well. So just a big thank you for taking the time to talk to us, and thank you everybody for joining in and for your questions.
Very nice discussion that we had. So thank you, Rosemary, for the invitation again, and it was nice to be here.
Thank you very much indeed. Right, and thank you everybody.